The aim of this video is to describe the main design features of a cohort study, discuss some of the potential biases, and describe its main strengths and weaknesses. So the basic features of a cohort study are that the exposure is measured at the start of the study when the participants are free of the outcome of interest, so we study a population at risk. The participants are then followed up to see whether those that are exposed are more likely to develop the outcome than those that aren't exposed, are unexposed. And the cohort is defined by some common characteristic. So here's an example. This is the Medical Research Council's 1946 British birth cohort study, and it's a long-running cohort study that recruited from all births recorded during 1 week in March in 1946. So that's the common characteristic defining the cohort. But one of the original aims of this study was to try to understand the drop in fertility rate prior to the establishment of the National Health Service, and the aims have since evolved over time, reflecting the socio-political and scientific interests of the time. So it's gone from studying child development and growth to its present form, where cohort members are now approaching their 70th birthday, and it's become a study of lifelong health and ageing. These sorts of lifelong studies with fairly broad and general aims are relatively rare. They rely on long continued investment from government and charities. So clearly, cohort studies do have the potential to be expensive and time consuming because of the follow-up period. However, cohorts can also be established using historical records. These are sometimes called historical or retrospective cohort studies. And in these sorts of studies, information on the exposure is obtained from pre-existing historical records, and subjects with a record are followed up for the occurrence of the outcome at any time using either other record sources or perhaps in the present time by contacting individuals. This has the advantage that the investigator does not have to wait for a long time for the outcome to occur. The term historical can be a bit misleading because it doesn't mean that the exposure information was collected retrospectively by recall. The exposure is obtained from pre-existing information. The term historical or retrospective refers to the fact that the cohort is established on the basis of this pre-existing information. A historical cohort study will thus have the defining characteristics of a cohort study, i.e. that the source population is a population at risk of the outcome. The Hertfordshire cohort study is an example of a historical birth cohort study. So in the early 1900s there was concern about the physical deterioration of British people, and in 1911 an inspector of midwives organised the transcription onto ledgers of information taken at birth and during infancy from health visits. And this continued until the NHS was formed in 1948. The information lay somewhat dormant until it was discovered around 1990 by a group of researchers who were interested in the early life predictors of adult health and disease. The researchers thus established a cohort based on these records and linked this information to mortality records from the National Health Service Central Register, and they were able to show the first link between birth weight as a marker of early life circumstances and later mortality from cardiovascular disease using data collected on individuals. Up to that point, evidence for what became known as the Barker hypothesis mainly came from ecological and descriptive studies of disease trends. So just to go back and mention a couple of additional bits of information on the cohort design. So the investigator controls the selection of the cohort, and the characteristic that defines the cohort does not have to be birth year, as in the two examples we've just described. It could be anything. For instance, we may establish a cohort by a disease characteristic. So for example, we could establish a cohort of individuals who have heart disease and track their survival longitudinally, recording various information on treatment and lifestyle as we follow them up. Here the outcome would be survival, and the cohort is at risk because all are alive at recruitment. Another example might be a cohort defined by their occupational status, an occupational cohort for instance. The Whitehall cohort is an example of such an occupational cohort, and that's a longitudinal study of London-based civil servants. In terms of the analysis of cohort studies, because we begin with a population without the outcome, i.e. a population at risk, if our outcome is binary, then we can estimate the incidence and risk of the outcome. So that's quite a nice feature. And it also means that we can use measures of association such as the risk ratio and risk difference. What about bias and limitations in cohort studies? So first of all, selection bias. Well, that's unlikely for a prospective cohort study in particular, because typically subjects are selected as a group of individuals before the exposure is actually measured. But loss to follow-up bias is something to consider, particularly when a lot of people drop out of the study or a large proportion of people drop out, meaning that their outcome is not established. And loss to follow-up bias will occur when the association between the exposure and outcome is different in the participants who were followed up, i.e. those that have an outcome, versus those who dropped out. So as an example, if we were interested in the relationship between socioeconomic inequalities in health, it might be the case that individuals from the lower socioeconomic groups that are also unhealthy are more likely to drop out of the study, just due to circumstances limiting their ability to participate. And if that was the case, we would underestimate the socioeconomic inequalities in health. In terms of measurement bias, random measurement error is something to consider for all studies. So if the measure of the exposure for each individual contains error, but this error is random or non-differential in the sense that it doesn't depend on the outcome, then this would underestimate any exposure outcome relationship. But measurement error can also be differential or can also depend on the outcome or depend on the exposure. And if that's the case, that might bias any association we see in either direction. And you'd have to think through how that might occur. And what about recall bias? Well, because the exposure information is ascertained or measured before the outcome, then that's not a problem for cohort studies. And of course, because it's an observational study, we have to consider confounding. And lastly, generalizability. Well, that would depend on the characteristics of the population studied as to whether the results of a particular cohort study can be generalized to other populations. Lastly, here's a list of the strengths and weaknesses of cohort studies, and it's not an exhaustive list, but again, we need to consider the context if we're critically appraising a cohort study, so it's not a case of just reading off these strengths and weaknesses. So, first of all, in terms of strengths, the exposure precedes the outcome, so that should prevent reverse causality bias. Selection bias is less likely with these sorts of designs. We can obtain a direct measure of incidence and risk, and we can study many hypotheses. And cohort studies are particularly good for rare exposures because we can select cohorts with a known high prevalence of the exposure of interest. So for example, if we're interested in an occupational exposure, we select an occupational cohort where a significant portion are likely to have the exposure. And we can measure exposures and outcomes over time, so hopefully we have more chance of capturing the relevant etiological window. And in terms of weaknesses, they're obviously time consuming and expensive, although maybe not so for a historical cohort study. And cohort studies are inefficient for rare outcomes. So if the outcome's rare, we'd need a really large sample size to capture enough people with the outcome. So for instance, consider studying an outcome that has an incidence of only 1% using a cohort study. Just to get 100 people with the outcome, we'd need to recruit 10,000 participants. And lastly, of course, confounding, always something to consider with observational design.